at like 10.
I'm saying about the ability to express. What ability to express? Now hers. Oh, no. It's just like,
Probably what he was talking about for like the lab. Yeah, for like how to do them. What he was explaining that was. You understood what he was saying. I was confused. No, at the beginning. Like the packet that we have to do for lab, and he was like showing us how to do one of them.
apologize to the people that
Yeah, so like I'm just like reading along, and then when I see like a point, I just kind of shortly silhouette it. And then at the very end, I did like a.
Yeah, I'm gonna go read the questions she put up on Blackboard. And I'm gonna cut it like yeah, I just wrote right now.
Questions?
I'm really slow readers. Don't have to read. They have to read the word. Just really laying down. I never really thought that. I mean, if that's what you want, however. I like getting straight to the point.
Yeah, just give us bullet points of like. Oh, that's right.
Yeah, and then I just like, sure. I was going to start a few pages tonight. Have you started it?
Not yet, but the word. Also parallelism, that word showed up in reading. Oh my gosh. This is basically bullet points of
Versus
like a
sorry, it's definitely not.
Why do you cite that word knowing that will give you so much better writing structure and still doesn't work?
Right. It doesn't do anything.
No.
I was trying to learn about the parallelism. Like, what are you saying right now? Like, what are you trying to tell me?
Yeah, that's kind of what I got.
Shady, there's like convoluted way to say it. Why don't you just tell me what you mean?
Yeah.
Like I was like, oh my god, I have 500 videos. Let me just take it up a step. I'm like, there's something really stuff.
1940. Also.
Okay.
Yeah, Wednesday 7, and we got like.
Yeah, like 4 weeks. Oh, that's why.
Oh, I did. Okay.
Yeah.
That's how I was looking at it.
I was like,
Yeah. So it's
Terrible.
like question after.
the rest of the site. I get like my actual sign. Your actual what? Bro, I didn't get like a registry page.
You're like, I don't know why it's showing up blurry. Well, you picked a bad picture.
Like all these pictures. Like not on the road.
That is for that. Well, that's. No, well. So like, I'm talking back twice as Greg, so like, oh, you're like, I hit that 2 times fall.
You're just like,
Yeah, I really have to like draw up my lecture listening to Stevie do like.
your lecture probably
And
let out your face.
Greg was like, actually,
Like
like the filler, like. And then like Peter's lecture could never. You know, the average and like judges.
All right, good morning, everybody. How's everybody's morning going so far? Not bad. Yeah, loving the cold weather, huh?
Getting better. Good.
Yeah, it is a little bit. I guess we can look on the bright side. All right, so just a little heads up. I apologize, lecture 2 did not properly record. So if you weren't here for that lecture, you're welcome to stop in my office anytime, Monday, Tuesday, or Wednesday after class. And I can, go through, answer any questions you have. It is going to be recording now. Just note that the first 10-ish minutes are just kind of this, so you can skip through those. And you can find the recording on Blackboard. It's in the file a folder that I've labeled Lecture 3. You can find the lecture and then the recording in there. All right, first, any questions, comments, concerns about anything we've gone through the last 2 lectures? No? Okay. That'll change before the exam. I'm just kidding. Okay, so lecture 3. So today we're going to be talking about microbial cell structure and function. We're going to be going through just about half or a few sections of chapter 3 in your book. So we'll go over the overview of what a bacterial cell looks like, membrane molecules and transport, and then the cell envelope and the cytoskeleton. So just a little overview, we're going to be talking about what makes up a bacterial cell. It's composed of, now we have some really cool techniques to be able to image bacterial cells and see, really pretty features inside of the cell. And so we see here, we have the nucleoid, ribosomes that are all this green all over the cell. And then this cell has an inner membrane and an outer membrane and a peptidoglycan layer. And so this would all make sense when we talk about the cell components and what their function is. But what's important to know is that most bacteria will share fundamental traits. There are differences in different types of bacteria, like gram-positive and gram-negative, but some of the more fundamental traits that they'll share is that they have a thick, complex outer envelope. The outer envelope is important because it's going to protect from environmental stress and just the environment in general, and it helps to mediate exchange from the external environment inside the cell. Bacteria also have a very compact genome, which means that there's little non-coding DNA. So if you compare that to eukaryotes, like us, for instance, we have a lot of non-coding DNA, just DNA that ends up becoming excised, they're introns, and so they don't code for any proteins. But bacteria actually have evolved to have DNA that codes for a lot of different genes that are translated to proteins with a function. And so the reason for this is that bacteria have this really sleek genome that can replicate quickly and efficiently so that the cell can actually divide and replicate. So beyond that, bacteria need to ensure that they have a high rate of reproduction. And so another component that contributes to this high rate of reproduction is having really tightly coordinated cell functions. And so bacteria are in this really complicated environment with other bacteria. They usually exist in a niche with other different types of microbes. And so there's this evolutionary pressure for bacteria to grow as fast as possible and outcompete other bacteria in that niche. So they have to be able to switch certain genes on and off and code for certain proteins in order to survive different stressors, whether it's from the environment, other bacteria surrounding them. So that's why they need to have these things in order to allow for this high rate of reproduction. So we talked about archaea, and like bacteria, they are prokaryotes. And so we talked about how they're understudied, and archaea are, we thought for a long time that, oh, they're just bacteria, but as of, I think it was the 70s, we started to learn more about the structure of archaea, and they actually resemble eukaryotes in some other ways, and as well as prokaryotes. So they're kind of like a mixed bag of features. So they have unique membranes, envelope structures, and specific gene expression machineries that make them different than bacteria. And eukaryotes, they have a nucleus and extensive membrane-bound organelles, which we've talked about before.
So an overview of the bacterial cell. Early in the 20th century, the bacterial cell was really presumed to be this like bag of soup where you just see like a circular cell, some like flagella on the outside, and then you have all these dots inside, which are the ribosomes, and then you have this DNA that's just kind of in the middle. But this is actually not, correct, and that's one of the beautiful things about biology is it continues to evolve and we continue to learn new things. And so modern research shows that the cell's parts actually fit together in an ordered structure. There's a reason for certain cell parts to be in a certain place in there. And so these cell parts are flexible and they're adaptable to changes in the environment. So this is a super resolution microscopy image that shows that this bacteria is really incredibly jam-packed with molecules and it's exquisitely organized. You see the DNA over here. And so the bacteria has to be able to turn on a dime when it comes to their expression profile of proteins that are necessary in that changing environment. Any questions at this point? All right. So the general modern model of a bacteria. So this is a gram-negative bacteria. This is just E. coli. And so just to show you some of the hallmark features, over here in the bottom center, we're looking at the nucleoid. And so you have all of this DNA. They're not membrane bound. They reside inside of the cytoplasm of the cell. And so it has, once you move outwards towards the cytoplasm, you start to see that there are different enzymes and RNA and ribosomes. Here we see the mRNA in these blue squiggles. And so that provides information for the ribosomes, which are in pink, that will make the polypeptide sequences that fold into functional proteins. I also want to note here that you see these little green things, so these are just a feature of the cells that kind of mimic what eukaryotes have in terms of just compacting the genome. And so we have histones that help to compact the genome, and so bacteria don't have histones, but the DNA isn't just loosely all over the place. There is some coordination. There are things that help to keep it tight and compact and make space for other things. And then if we go all the way out to the outer cell envelope, here we see an outer membrane, and then we see an inner membrane. And so this is because it's a gram-negative cell, it's going to have these two membranes. So the inner membrane we call the cell membrane, and then in the middle, in the periplasm, we have the phospholipid bilayer, and we'll talk a little bit more about that. And so over here, there's a bunch of these sugar strands that are connected by peptides. And so out of the envelope, there's this lipopolysaccharide, or LPS. These are sugar strands, and so they're lipids with really long chains that help to evade our immune system. So they're outside of the cell wall. So we have our cell wall to protect the cell, or not we. Bacteria have the cell wall to protect the cell, but then they also have these LPS. So this is in the gram-negative bacteria. We also have this flagella right here. So this is, there's this motor that's embedded within the flagella, and then this turning of this rotor helps the cell propel and move. So some general features. And so understanding the composition of bacteria, this is a fairly new field. There was this this professor, Dr. Fred Neidhart at U of M, actually, that studied a lot of this, and some of his work basically came up with the notions that all cells share common chemical components. And so that contains water, there's essential ions, small organic molecules, such as lipids, sugars, and then there's other molecules like macromolecules like DNA, RNA, proteins. But the important thing to remember is that the composition of every bacterial cell will vary with the species of the bacteria, the growth phase, so how fast or slow it can grow, and the environmental conditions. So they also have to be able to change on a dime, like I said, and change their composition in order to survive. So this is just a figure. This is from his work and others, just kind of giving you an idea. You don't need to remember any of these percentages, but giving you an idea of like the molecules of the bacterial cell consists of about 70% water, 16% proteins, about 6% RNA molecules, and lipids are like a small percentage, as well as DNA, it's about 1%. You don't have to remember any of these, but just to give you a general idea, and this is also found in your textbook. So we talked about just some of the general components of bacteria. And so here we have the cytoplasm, and this is where all of these different enzymes, ribosomes, DNA, all of these molecular molecules that help life function are inside of the cytoplasm. And so this has to be protected and contained. And so the thing that protects it is the cell membrane. And so we'll talk a little bit about the cell membrane. And so it is the structure that defines the existence of a cell. So what that means is it helps to define itself from non-self. So the cell membrane helps to keep things outside in the external environment and bring things in that are necessary. So it helps the bacteria really identify what is self from non-self. So it's a really important structure. And so it contains the cytoplasm, and it also mediates exchange with the external environment that you see here in the blue. So the cell membrane consists of a phospholipid bilayer, which I'm sure you guys all have heard of in terms of just your general biology courses. So you have your phospholipid bilayer with your hydrophilic heads and your hydrophobic fatty acid chains. And then you have these membrane proteins that are embedded within the cell membrane. The hydrophobic fatty acid chains are directed inwards, so they are not facing the external environment or the cytoplasm, and that's because they are hydrophobic, which just means they're hiding from water. They don't like water. And the phospholipid heads face each other inwardly, and so these are molecules that are polar and can be facing the extracellular environment. And so one other thing I want to mention is there are also this structure here. This is something a little bit newer that researchers have discovered. There are these hopanoid molecules that is essentially a stiffening agent that helps stiffen and maintain the stability of the membrane when the membrane can be destabilized. So sometimes there are, like I said, environmental things that can occur that will change, that will make this cell change its composition. And so one of those things is if the cell is experiencing a really hot environment, hot temperatures destabilize proteins. And so the cell membrane will start wanting to destabilize, and so one of the mechanisms that the cell has is it starts to produce these hopanoids, these stiffening agents, to help kind of maintain the rigidity and the stiffening of the cell membrane. So in terms of content and actual surface area, there's a lot of proteins in this cell membrane. Any questions on this? So yeah, essentially about half of the cell membrane consists of proteins. So that's just something that to know in terms of like the content and the surface area. So what do these proteins do, these membrane proteins? They have multiple functions. They can serve as structural support for the cell. They can detect environmental signals. They can secrete virulence factors and communication signals. So there's this concept of chemotaxis, which is the detection of environmental signals, and the cell can either move towards or away from that signal depending on whether it's going to be beneficial or harmful for the cell. And so membrane proteins serve that function. And then they are also very important in transporting ions and other things across the cell membrane and storing energy. So let's talk a little bit more about membrane proteins transporting things across the cell membrane. So we already said that the cell membrane acts as a barrier to keep water-soluble proteins and other cell components within the cytoplasm. So it's protecting the cell from things that are non-self. So how do nutrients get in, toxins get out? We have specific membrane proteins here that are necessary for selectively transporting those molecules. So polar molecules and charged molecules require transport through specific protein transporters. And the two types of protein transport is passive transport and active transport. And so just briefly, I'm pretty sure you guys have covered a little bit of this before, but if not, just to briefly put this into context, passive transport, the molecules will be moving along their concentration gradient, or they're moving down their concentration gradient. So there's usually a high concentration in the extracellular environment and a low concentration of that thing inside of the cell, and so it's going to go from high to low. Active transport will require energy. Passive transport doesn't because it's moving along its concentration gradient. But with active transport, the molecules are moving against their concentration gradient, so it requires energy. So there are a number of membrane proteins, and just like the phospholipid bilayer here, the heads and the tails, the membrane proteins also have hydrophilic and hydrophobic regions that help lock it into the membrane. So these charges are really important, not just in the phospholipid bilayer. So there are hydrophobic residues that are implanted into the hydrophobic region. So you can see here they're white, so neutral charge. And so they're not charged as much, and so they hide out here in the hydrophobic region of the membrane. And then there's like amino acids that have electrostatic charges that are exposed to the extracellular environment and the intracellular space over here. So they're either positively or negative charged, as seen here in the red or the blue. So now we're going to talk a little bit more about these phospholipids and some of their functions. And so fatty acid chains, they can be unsaturated or saturated. So one of the things to keep straight is unsaturated means that there are more hydrogen atoms that can be added to the hydrocarbons to make it saturated. So basically what that means is it's more fluid. You can add more hydrogen atoms to make it more stable. And one other way to remember saturated, I think solid. So SS, saturated, solid. Saturated things are like butter, unsaturated is like liquid oil. And so at room temperature, that is. And so in order to function, these membranes have to be fluid. And so bacterial cells modulate the composition of their membrane in order to maintain that fluidity. So they're able to change the composition of those phospholipid heads and the fatty acids inside. So you don't want to be too fluid because that will destabilize the membrane. So you have to find this Goldilocks zone of maintaining membrane fluidity without actually killing the cell. So here we have let's look at a couple of these. So if we're looking at unsaturated, so this oleic acid and cis, basically it is more fluid, and you can tell this because of this bond over here that makes it have this kink. And so kinks like this allow the fatty acid to be more fluid, and so kinks further increase fluidity. If you have something like this, that's a cyclic structure, that makes the fatty acid more straight, and so that decreases fluidity. So it's important to remember that a kink will bend it and it'll make increase fluidity, and cyclic structures like this decrease fluidity. So here we have oleic acid as well as over here. So this one is in cis, this one's in trans. So the trans version, I'm not going to go into the chemistry of this, but essentially when oleic acid is in trans, it's more saturated, so it's less fluid, And then when it's in cis, it's going to be more fluid and it's unsaturated because it has this kink. So the cell can go back and forth and change some of these features depending on whether it wants to be more fluid or it needs to become more rigid. Does that make sense? So let's give an example here. If we have a cell and we put it in an environment that increases, where the temperatures increase 10 degrees, is the cell gonna make fatty acid molecules and change the composition of that to be more fluid or less fluid? Do you think they'll want to have more kinks or less kinks if it's in a hot environment? So what does temperature do to the cell? It's going to destabilize things. So it's going to start to become more fluid. So if a cell is all of a sudden in a really hot environment, it's going to want to change its composition so that it is going to be, maybe if it's in the cis version here, it'll change itself to be in the trans alignment here, and that way it can be more rigid and less fluid. So like I said, the cells can also synthesize these haploid molecules by embedding them in the membrane and by crowding, so they limit the motion of these tails. So another example is, if a cell is in a very hot temperature, it can change the kinks in the fatty acids, or it can start to maybe put in some of these haploids and synthesize some of these haploids, and they're embedded in there so that they allow these fatty acids to not be flailing as much and be so fluid. Yeah. You have a question?
Yeah, so cells, they have the capacity to make kinks or take them away, and is that what changes fluidity, or is the only thing that changes their fluidity the external factors such as temperature? That is what directly changes the amount of kinks.
Great question. All of the above. That's all correct. So temperature will change the fluidity because it's destabilizing the proteins within the cell. But then as a response mechanism, the fatty acids can then the cell can then synthesize fatty acids that have more, less kinks or more kinks, so both of those can happen. So there's different ways to change the fluidity. So it can be from the external environment, but then as a defense or response mechanism, the cell will kind of want to reverse that and make sure it's in that Goldilocks zone, just being happy and not too fluid because of that temperature increase. And then vice versa, if you put it in a really cold environment, it's going to become more rigid. So that is influencing the fluidity, but then the cell can go and make more kinks in their fatty acids, and that will give it a little bit more fluidity so that that cold temperature doesn't affect its survival and growth. Yeah.
So the cell is just working the whole time to maintain homeostasis?
Yes, yes, always, yeah, exactly. Yeah.
Is there a reason outside of temperature and salinity that those would change?
Yeah, absolutely. That's a really good question. So there are a lot of other factors like pressure to the cell, competition for nutrients, things like that. Mostly it's going to be something that's going to, if it's going to destabilize the cell wall or the protein, or if something's going to attack it, there's always a defense mechanism. Like Hannah said, it's always trying to be in this homeostatic environment, and that's why it has a lot of these genes. It doesn't have a lot of non-coding genes because it wants to keep genes that can code for proteins really quickly, and they replicate really fast, so that's why they're able to survive those things. So they're different from eukaryotes in that way. And microbes really evolved on this planet first, so they really know how to maintain their homeostasis and to survive and combat a lot of things. So they're really smart, and we think of them as microscopic and we can beat them, but really they have a one-up on us. So they're really interested in the things that they can do to maintain their survival and persistence. Does that make sense? Those are all really good questions. So a concept check. compared to a microbe in our gut, the phospholipid bilayer of a microbe living in a hot spring is more likely to have A, lipids that provide greater fluidity, B, more unsaturated fatty acids, C, less cyclization of their fatty acids, D, more peptidoglycan, or E, lipids providing greater membrane stiffness. So this is the type of question that I would ask on an exam. And so here I am throwing in all the concepts of fluidity, unsaturated, like having the cyclization, and the cell wall components like peptidoglycan, and then lipids providing more stiffness. So you can think about that a little bit. If you think about it, microbes in our gut versus something living in a hot spring. So hot spring, the difference between microbes in our gut and hot spring is temperature in this case. So any guys' thoughts as to what you think it is, or maybe if you want to, yeah.
I thought it was E.
Yep, exactly, it's E. So A through C increase membrane fluidity. So we don't want that for the microbes living in a hot spring. So A increases fluidity, B, having more unsaturated means more fluidity. C, less cyclization also means more fluidity. Remember that if there's cyclization of the bonds, it's gonna keep it stiff and it's more rigid. More peptidoglycan, that has to do with the peptidoglycan layer, so that doesn't have anything to do with the phospholipid bilayer. And then having more stiffness is the correct answer. So yeah, everybody, makes sense to everybody? Okay. So we're going to talk a little bit about the cell wall and some of the outer layers. So prokaryotes, they protect the cell membrane with this exoskeleton. So for most species, it's the cell envelope, and it includes at least one structural supporting layer. And the most common structural support is the cell wall. So that's just a long way to say that the cell wall is what protects the inner part of the cell. There's a few prokaryotes that have a cell membrane with no outer layers, just because there's an exception to every rule, so I have to put this up for you guys, but we're going to focus more on what the cell membrane does. So let's look at up here, we have a gram-negative cell, and so we're going to talk a little bit more about the difference between gram-negative and gram-positive cells and their cell walls, because that's going to be important for lab next week when we do the gram. But for now, I'm just going to show you this image where you have the gram-negative cell, and you have this inner membrane, and then a thin peptidoglycan layer, and then this outer membrane right here. Whereas in gram-positive cells, you have the cell membrane and then just this really thin peptidoglycan layer. This is all the peptidoglycan layer. And so this is essentially the cell wall versus having the inner and the outer membrane. So the cell wall is a single molecule. It provides structural support for the cell. It helps to confer shape and rigidity. Another term for it, when it's a single interlinked molecule, is a sacculus. And so it really becomes this cage-like structure that's highly porous, so it allows ions and organic molecules go in and out. And there are these glycan strands, and so they're labeled G or M. So G is here referring to N-acetylglucosamine, and M is N-acetylmuramic acid. And you don't have to remember these long names, but you have these like colorful dots in the middle, and these are amino acids that cross-link and connect these strands together. And so that's why it's interweaved, and it's really strong. It's a single molecule because of all of these bonds. So I do want you guys to remember what the peptidoglycan molecule consists of. So the cell walls are made up of a peptidoglycan layer. And so this is actually an image and a schematic that I'm going to show you later that I've created to explain, the cell walls of bacteria. And so over here, I'm showing you the phospholipid bilayer as we talked about. And then on top of that, we have the cell wall. So if we zoom into this part, so that's what I'm showing you right here. The cell wall is made up of these glycans or these sugars, so two different types of sugars. So NAG and NAM is how I'm referring to them. And so you have these bound to each other, and they're in these darker purple dots here. And these polymers now, because they're bound to one another, they are linked by probably about 4 to 6 different amino acids. And there are these cross bridges that are formed between the amino acids that help to connect the parallel glycan strands. So it's all connected. And it's important to know that the amino acids are what are making that cross bridge to link the parallel strands of these sugars. So they're bound to the amino acids of the next layer and the next layer and the next layer. Lastly, we have this transpeptidase enzyme, and this essentially clicks the amino acid groups together, allowing for the cell wall to be rigid for the bacteria. So this also helps to cross-link the peptidoglycan layer. So two things that help click the whole peptidoglycan cell wall is the amino acids and this transpeptidase. So this image is in your books, and so this is just showing you the actual chemistry of what these look like. You don't need to memorize this, it's just an FYI slide for you guys. So we're talking about bacteria here, so let's zoom out a little bit. So when we're talking about bacteria, there are bacteria that can cause disease and harm us, and so when we're trying to create antibiotics to help kill these certain types of bacteria or help to evade infection, we target things that are a part of the bacteria but not a part of us, because we don't want to target something that we have in common with the bacteria, because then it'll be kind of attacking yourself. So we take advantage of the bacterial cell wall and its components when creating antibiotics. So peptidoglycan synthesis is a target for antibiotics. How many of you guys have taken like penicillin, ampicillin, amoxicillin, Peniton? I'm sure probably majority of you, if not all, at some point in your lifetime. So all of those are a penicillin type of antibiotic. And so because peptidoglycan is unique to bacteria, there are enzymes that are part of peptidoglycan's biosynthesis that make excellent targets for new antibiotics. For example, this transpeptidase enzyme, this thing that cross-links the peptides, is the target of penicillin antibiotics. So basically, by adding antibiotics, we're interfering with this being able to click and link this whole structure together. So in other words, we're targeting the cell wall of the bacteria. We're destabilizing the cell wall with certain antibiotics. So I'm going to show you the mechanism of action of how a penicillin antibiotic works. So these penicillin antibiotics are part of a class of antibiotics that are called beta-lactam antibiotics. And so this is the core structure of penicillin. And so the commonality between penicillin and then all the other penicillin type antibiotics is they all have this structure here. That's the beta-lactam ring. So this ring, essentially, I think it looks like a house. So I've drawn a schematic where I've made it look like this sort of house structure. And so beta-lactam antibiotics are bactericidal agents, which means that they actually kill the bacteria. And they do this by interrupting the bacterial cell wall formation by binding to this transpeptidase. So penicillin, so this beta-lactam ring in penicillin will bind this transpeptidase, which then disrupts its ability to cross-link this peptidoglycan layer. And once it does this, there's an opening in the cell wall. It's not clicked together, and so water will rush into the cell, and the cell wall falls apart, and it lyses, and it bursts, and the cells die. So that's how certain penicillin antibiotics work, or beta-lactam antibiotics. Does that make sense? Okay. So, unfortunately, I'm sure you all know that the widespread use of antibiotics selects for evolution of resistant strains. Bacteria are smart. They've been around for a very long time. They're going to continue to be around, and that's because they are able to combat evolutionary pressures. And so one of those things is antibiotic pressures. So there's a lot of antibiotic resistance that can occur. And so I'm going to talk about two types of mechanisms of resistance that bacteria can incorporate into their genome in order to combat the antibiotics killing them. So the first one is bacteria is circular. The DNA is circular. It's double-stranded. So over here I'm showing you a cell. Here we have the bacterial DNA. So it's going to contain nucleotides that will create certain genes, and one of those genes is going to be the transpeptidase that it's going to need to click the amino acids. So there could be a mutation that occurs in the transpeptidase that changes this gene to code for a transpeptidase that's shaped slightly different. So instead of it being squared at the ends, it's more rounded. So it still produces a transpeptidase, but now it has a mutation that changes the shape. So now when the beta-lactam ring comes in and tries to bind the transpeptidase and destabilize it, so when you try to use the antibiotic, it's going to go in and it's not going to be able to bind. So the transpeptidase can still cross-link these amino acids, but the beta-lactam ring of penicillin can no longer bind. So it changes the shape of this transpeptidase to evade our antibiotic. Does that make sense? So this no longer can bind. But it's important to note that the transpeptidase still works, even if it's shaped differently. But now our antibiotic doesn't work. Does that make sense? Any questions?
Yeah. So like, obviously, say you have like antibiotic resistant strep, try the antibiotic, doesn't work, so your doctor typically gives you like Z-Pack. So essentially the structure of the Z-Pack is just accommodating for the mutation of.
Yeah, exactly. So so there's different classes of antibiotics, and there's different types of antibiotics. So if they know that you have a resistant strain of something, then they will typically want to give you an antibiotic that doesn't target the cell wall. They'll give you something that targets a different part of the bacterial cell because they know that it's not going to work because you're resistant. So there's different types of antibiotic classes that target different parts of the bacterial cell, and that's part of us trying to evolve and keep up with this resistance. But yeah, that's also a big issue in hospitals and ICU settings where critically ill patients get combinations of different antibiotics, and so that helps bacteria become more resistant. And I'm not going to go into all of those fun things, but if you want to talk more about that, you can come to my office. But yeah, absolutely, like that's why doctors will switch up on you different antibiotics when they feel like things aren't working. Yeah.
So you're saying the transpeptidase change shape and it still works. Does that mean like the amino acid chains have to change anything too, or they just still work?
Nope. Yeah, no. So the amino acids aren't being targeted. They're still being able to link together and form the parallel strands. They're able to still cross-link, but then we need the transpeptidase to click together these amino acids. So over here and here, you see that they're not clicked together. So in order for all of this to be clicked together, they need the transpeptidase. And so the beta-lactam ring of penicillin will come in and disturb this, but in this case, it's evolved to change its shape, so it still clicks them perfectly. Amino acids are still the same. It's just a difference in shape to evade the antibiotic. So. Good. Anything else? So this is the first way. The first way is it causes a mutation in the transpeptidase enzyme that will cause it to change shape. The second way of resistance that can occur is bacteria share genes. So we know that they have specialized traits that allow them to jump ship, have their genes jump ship from one species to the next. So let's say that there's another bacteria around it. So here's our bacteria with the transpeptidase, and then there's another bacteria in the environment that has this enzyme called beta-lactamase. So beta-lactamase just means that it can break down beta-lactam. So one way to remember that is anytime you have ase at the end of something, it means that it's an enzyme and it breaks down something. So beta-lactamase breaks down beta-lactam. So bacteria can share genes. So when these bacteria come together, they can share this genetic material. And so here we're looking at this genetic exchange of the beta-lactamase. So here in the green, it's being shared and incorporated into the genome of our first bacteria. And so when this happens, the gene is inserted into this bacteria, and then it produces an enzyme that's called beta-lactamase. And so here I have this as a Pac-Man schematic to remind you that it's eating away at something or it's breaking something. So this beta-lactamase likes to break down beta-lactam, which means that if the beta-lactamase is being produced, it's gonna break down this beta-lactam ring. And then when the beta-lactam ring is broken, the penicillin can no longer bind the transpeptidase, and it doesn't inhibit the cell wall formation and kill the bacteria. So beta-lactam ring no longer works, and it no longer fits inside of the transpeptidase, so it can't break down the cell wall. Any questions on this? So genetic exchange is the second type. And beta-lactamase is probably one of the most common types of antibiotic resistance, where these bacteria end up encoding and synthesizing this beta-lactamase enzyme that directly goes after our antibiotic and breaks it down so that it can no longer function appropriately. Any questions?
Is there any medicine that can knock down that enzyme, the beta-lactamase?
It's a difficult, I don't know for sure. I don't know, like honestly on the medical side of things, I'm sure there have been attempts and ways to combat resistance, but I'm not exactly sure the answer to that. So yeah, the transpeptidase no longer works. Or sorry, it works, but the beta-lactam ring no longer fits. So, okay, we're gonna take a step back from the cell wall and talk a little bit more about the actual envelope layers that provide the structural support of protection. So, the envelope composition defines whether it's a gram-positive or gram-negative bacteria. So the gram-positive bacteria have a thick cell wall, and that means that there's about 3 to 20 layers of peptidoglycan. Gram-negative bacteria have a very thin cell wall with about 1 to 2 layers of peptidoglycan. So to show you an image of this, I really like this image in the textbook. So gram-positive cells, over here you see the cell membrane, and then you have this really thick peptidoglycan layer. And then I want you to pay attention here to this teichoic acid that is linking, helping to make more like rigidity within this peptidoglycan layer. And then there's this S layer, and then there's these glycosyl chains that help to protect it from environmental things. In the gram-negative cell, we have this inner membrane, so the phospholipid bilayer, and then we have this periplasm with just like 1 or 2 peptidoglycan layers. And so these, because there isn't so much of the peptidoglycan in here, in order for it to attach to this outer membrane that these cells have, there are these lipoproteins that connect it. And here we have the second outer membrane, and then we have these LPS or chains that help protect it from environmental stress, and this S layer. So actually in the 5th edition of this book, so the 6th edition is the most recent, and the 5th edition of this book, this S layer on gram-negative bacteria wasn't there in the schematic, so it was updated because we recently found out that gram-negative cells have this S layer too. So last year, professors were telling students that gram-negative cells don't have this S layer, so it's always evolving, always changing. So yeah, the cell wall here in the gram-positive bacteria is threaded by a molecule called teichoic acid. And it's found that this teichoic acid is what helps hold on to the crystal violet when it's in complex with iodine in the gram stain. So threaded by teichoic acids, these are negatively charged that are what help the cell retain the gram stain. So the gram-positive cells being purple, that's why, because the iodine complex with the crystal violet helps to lock in the dye via these teichoic acids. There's also these types of capsules that are made of glycoproteins and polysaccharides that helps to protect, so that's this part of the cell, that helps to protect it from the outside environment, from phagocytosis by macrophages. So these are really slippery and they help keep the cell from getting sucked in by macrophages and immune cells, and also helps keep them from drying out. So now we know that these are also found in gram-negative cells. So it consists of both the S layer and these glycosyl chains. And so they're sticky, they help the cell not dry out, and they can be seen in this case here, we're looking at it via negative staining or dark field microscopy. So this is the gram-positive cell envelope. Gram-negative cell envelope, to beat this home, we have the thin peptidoglycan layer, and so we have the inner membrane and the outer membrane, and so the outer membrane helps to confer defensive abilities and toxigenic properties in many pathogens. So because it doesn't have this really thick peptidoglycan layer, it has this outer membrane for further protection. The outer facing leaflet, so out over here, we have these lipopolysaccharides, or LPS, and so these help to cross-link ions, which I'm not going to get into the magnesium here, but there are also these porins in the outer membrane that are very large diffusion channels that help things to go in and out. So LPS is part of this outward-facing leaflet, and then in the inward-facing leaflet, like I mentioned, there are these lipoproteins that connect to the outer membrane of the cell wall. So this is gram-negative bacteria. Any questions at this point? Okay. So just talking a little bit more about the S layer, it's a protein coat that's common across bacteria, we now know. It's just an additional protective layer that's commonly found in bacteria and archaea. And so if we zoom in here, we see that it's a crystalline structure. It's just these proteins, and it's really thick and crystallized. And it helps to contribute to the cell shape and protect the cell from osmotic stress and drying out. So it's rigid, but it's still porous enough to allow things to pass through. So for the last slide and the last concept check, which of the following is not a component of the peptidoglycan layer? So N-acetylmuramic acid, or NAM, N-acetylglucosamine, which is NAG, lipopolysaccharides, amino acids, or peptide crosslinks. Peptidoglycan, you can go to the purple image, the schematic that I had created, which is not a component. Yep, C. LPS, because LPS, as I said, is on the outer leaflet of the outer membrane, not the cell wall. So LPS is over here. It has nothing to do with the peptidoglycan, and then similarly here, it's going to be in this area and not in the peptidoglycan layer. So with that, before you guys head out, I know you have to go for your thing, but I want to give you guys, before you leave, this worksheet that is gonna, it has some questions on here to review concepts from lectures 1 through 3. So you guys can go ahead and work on this on your own time. You're welcome to stay until the end of class now, or you can work on this and then bring it back with you on Monday. And Monday we'll go over them and we'll make sure that you guys have all your questions answered. Since I am not holding office hours officially, this is a good way for you guys to review the material and keep up and ask questions. So if you guys can take one before you leave, and then, yeah, so doing it and participating will give you points for this, so just an FYI. It's not gonna be graded, but we'll go through it together. If you've done it, I'll give you the points, yeah.
For the gram positive, I can't there, but I didn't see where you said the LPS.
Yeah, so it would be part of what I labeled the glycosyl chains.
Oh, okay.
Yeah, yeah.
Anyways, I have to go bed for this.
Yeah. Yeah, let's do it. I think I drank all that already. That would have been the second time I saw
Like that 35, so we'll take some.